So we're sort of halfway through our workshop at this point, and I thought that it might be a good point, which I think we've already started, to kind of start thinking about the discussion, and we're going to have one this afternoon. So maybe to take a step back and just start to think big picture, what are we doing here? So I thought I would just start a little bit by thinking about this question, what is it that we actually want to know? Like, why are we here? And how will a connectome help us know it? And depending on the perspective you come from, these thoughts that I have are probably either obvious, naive, or controversial, which seemed like a great place to start for a discussion. So again, kind of stepping back and taking like the 10,000-foot view, what do we want to know? Okay, obviously that's a question that's different for every person. When somebody asks me that at, like a party, I usually say something like, I want to know how the brain works. Or, I want to solve the brain, which obviously it makes no sense whatsoever. That's kind of a contentless answer. But okay, let's dig into that a little bit. What might it actually mean to, quote unquote, solve the brain? Again, at a kind of very gross level, I basically am interested in understanding how these kinds of electrical dynamics, right, in the cells in our heads lead to these kinds of complex behaviors, right? So how can we understand how to go from there to there? And so what would it mean to solve the brain? Might that mean that we could in some way actually predict sort of the output of the system? If we could predict the behavior, would that, check, we've done our job? Or from certain perspectives, it might be that we would actually be able to cure some kinds of mental illness, some kinds of disease. That might be the goal. Maybe the one I'm a little bit closer to is this one, could we consider having solved the brain if we could actually articulate some collection of principles or mechanisms that actually produce the perceptual and behavioral phenomena that we observe. Okay, that itself is also vague. You're welcome us to dig into what that means. It kind of leads us to this part of the thought process of what does an answer actually look like? And I would actually just suggest that we as a field or as fields don't spend enough time talking about this, like what actually form of answer do we want? What would be satisfying? To some people, it might be a very, very thick textbook. I think textbooks enumerating principles of neuroscience are growing in size. Or it might be ChatGPT, right? If we can model it, are we done? Like, do we understand it? I kind of put this out there to kind of bring in the mathematicians, hopefully, is it just a set of equations? What if we could actually put some kind of closed form equation on every edge between every node in these connectomes, right? Is that even achievable? Or, maybe not every edge, but maybe every sort of little microcircuit. Might that be something that we could actually think of doing as we try to understand these phenomena? Okay, and then of course, an important thing to engage with is, where are we stuck? Why have we not solved this yet? What is holding us up? And when I think about this, there's one aspect of this that kind of blows my mind, which is that we actually basically already know all the parts and how they work, right? So we are not at the point of like Louis Pasteur trying to figure out where disease comes from and like looking down Van Leeuwenhoek's microscope and discovering germs, right, and bacteria. Like, there's probably not a whole new phenomenon to discover in terms of biological phenomena. Maybe we're more at the point of Watson and Crick, right, where they had all of the pieces, and actually the missing part, the magic of how it all works, was the structure, right? How do those things fit together? And that's where I think, we really want to reach across disciplines, because that structure might be something that doesn't exactly look like biology, but could be this kind of the missing part to understanding how the magic of the brain works, as it were. Okay, and so why is this hard? Well, you've heard lots of reasons, I think, already throughout this talk, just to kind of highlight two of them that keep me up at night. We not only do we have all the parts, but like now we have connectomes. Let me bring it back to connectomes. Like, not only do we know basically, how ion channels create action potentials and how neurons talk to each other by synapses, both chemical and electrical. We now know more about mitochondria than I used to know like an hour ago. But yeah, now for some animals, we actually know how those are all literally linked up anatomically. But what is still hard about this? Well, the parameter space is really large, right? Like, in theory, we know all the parts, but it's true that we don't actually know the resting membrane potential of every neuron in our head. Like, do we actually need to know that level of detail? Or can I know, just kind of from basic biophysics, I know the range of action potentials that are, or resting membrane potentials that are possible? Like, is that sufficient? But obviously, the parameter space is large enough. There's so many combinations. We're probably not going to just be monkeys typing on a typewriter and like come up with the solution of the brain accidentally. And then the other one, and this is one, again, that I think we don't engage enough with, is that the answer is multi-scale. And I think Srini did such a great job of introducing this, with his intro. So, if you maybe kind of harken back to that. But not only are we trying to understand something across scales, right? So we're trying to understand how something in the properties of individual neurons, or maybe even individual ion fluxes, is relating to like emergent behavioral phenomena. But the answers to every little kind of behavioral phenomena or computation question are also at different scales. And so, today, mostly what I'm going to talk about for you are like a couple examples from the fly, one of which comes down to, spike timing, but we've heard other solutions that come down to like larger network properties, right? So the answers aren't all at the same scale. And I don't think it will necessarily be satisfying to just decide as a group, okay, this is the level of abstraction that we're going to be happy with. We're going to, threshold at 5 synapses, and then we're going to accept nodes and edges, with these properties. Like, right, there's probably no one answer to that question. So how do we deal with some kind of like heterogeneous type model or solution or approach that's really going to give us the whole answer? Okay, so that's a little bit of something about what we want to know, and again, hope that we can sort of engage in that discussion more later this afternoon. So then bringing it back to the connectome, kind of the theme of this meeting, like how will a connectome help us know it? One thing that's maybe not so direct to the answer, but I think is important, is that connectomes are democratizing. So right now there's a lot of information in the hands of a lot of people, and it's clear that it's going to take many different perspectives to come up with a solution. So I really like that now there's widespread access to data. Connectomes let us think on a brain-wide scale, and I'm going to show you a specific example of that in a second, but just to cartoon it, right? I think there's a lot of neuroscience that looked like this before. It was the whole finding your keys under the lamppost situation where especially biologists would like to think of particular brain regions, and what that particular brain region does, when we really know that a lot of computation is widespread, or at least there's a lot of redundancy or degeneracy or things that happen more on this scale. And so now having connectomes, or maybe focusing on animals like the fly, where you can kind of look globally, it lets us actually kind of create a whole different picture and perspective. Okay, connectomes very directly enable a different approach to experiments, and I'll talk to you a little bit more about that. We can actually just ask questions to the biology and make inferences that we couldn't before, and therefore they reveal new functional motifs. And then finally, connectomes can really be a bridge between theory and experiment, or between model and behavioral phenomenon. And the hope is that, right, by coming together, that speeds conceptual insight. Okay, so let me dig a little bit into the biology I do to hopefully illustrate a couple of these and just kind of make concrete some examples of how, from a biologist's perspective, I use information like the connectome, at least at a kind of small scale of the fly, in order to try to understand something about how the nervous system generates behavior. So this is the model organism I study, and this is the situation that I spent about 10 years thinking about: flies getting eaten by predators. And most predators that eat flies, happens to be our carnivorous caterpillar, will attack them and create a visual looming stimulus on the fly's eye. Flies are highly visual animals, just like ourselves, and so this is a sort of a dark shadow that expands rapidly on the fly's retina. Now, flies are not the only animals subject to predation, right? This is one of the things that I like about studying these kinds of innate behaviors, is that there's sort of a long evolutionary history across many organisms that have faced similar challenges. And just one thing I like to point out about where the connectome has helped a lot is that if we look at neural architectures for rapid sensory motor responses, and particularly escape responses, for maybe the last 50 years, this is really the picture that I think people have had, especially in insects. And this is a really famous example from the locust, where, in 1974, Mike O'Shea discovered this LGMD neuron, which is a looming detector. It's an amazing looming detector, this wide field neuron. And we know that it synapses directly on this other neuron, which is a descending neuron that triggers the locus to do an escape jump. And so then I think this is then the conceptual picture that a lot of people have that, oh, okay, maybe at least in invertebrates, you basically have very specific ethologically tuned detectors that are linked up to these motor pathways that do a single thing, and then this is how you're going to build an organism. And maybe even the frog's retina is shaped this way, right? Frogs have little sort of worm detectors. Okay, but instantly, once you have a connectome, and if you have a little bit of functional data, it's clear that, that this cartoon just has nothing to do with reality, right? We don't have these pairs of like very specialized receptors, these very specialized responses that are the whole story. Because in the fly, we can immediately see that right here in red, I've colored some of the fly's looming detectors and then some of the descending pathways they connect to. And right already you can feel like this feels more like a brain, right? Like even in an animal like the fly that has many orders of magnitude fewer neurons than us, right? It's very complex immediately. And what's more, we can see not just kind of these individual microcircuits, if you will, but whole pathways, right? So here's the fly's brain. These different colored areas are sort of different regions of the fly's brain, different neuropil. And already from the connectome, we have a very good coarse idea of how all visual information flows into the fly, right? So I don't have to just talk about looming responses to a particular predator. I can actually look at all visual pathways and try to understand if there's specialization amongst them or actually there's some kind of distributed processing. So for example, let me go to this slide to show you this. There's 3 major visual pathways from the optic lobe into the central brain of the fly. This will surely be refined further as we dig into the connectome more, but kind of broad strokes. One, you've heard a really nice story from Vivek and Larry about, which is the pathway that goes through the central complex and then eventually will connect to through the lateral accessory lobe to descending neurons that go into the fly's nerve cord. The other has been touched on and of course we heard that that's role in navigation and these wonderful head direction neurons. The other pathway that we've maybe touched a little bit on, but actually not so much in this meeting, is one that goes to an area called the mushroom body, which is the fly's associative learning center. And that takes sort of very primitive visual signals, so color and things like this, and brings it up to the mushroom bodies. And then the third pathway, which is the one I'm going to talk to you about today, is one that goes from the optic lobe into these very nearby ventrolateral neuropils, and then kind of sort of immediately down into descending neurons. And as I alluded to with the video, I am going to talk to you about rapid escape responses and looming detecting neurons. And just to say, even early at this point, that while I'm going to tell you a whole story about how that occurs in this ventrolateral pathway, we know that there are looming responses in neurons in the central complex, right? So again, just kind of having in the background of our minds this global view, however much detail we learn about an individual pathway, there's probably some kind of global processing important. Yeah?
I wanted to clarify the pathway notion. So
yeah.
Related to the previous kind of like picture of like a feed-forward or some kind of like one pathway, here you mean by pathway like a more complex network, but still you have those pathways that are kind of unidirectional.
Yeah, that's a great question. And actually, so let me answer it and show you this next slide. So we have this same question. So right, there's this notion that at some point there has to be some kind of feed forward of information from the photoreceptor to the motor neuron, right? If we're going to see something and we're going to move. So in that sense, there's progress to the brain. But we also heard lots about recurrent networks and these loops, and so how do we reconcile those two views? And I don't have the full answer, but we wanted to turn to some of these different pathways and areas to at least get some kind of coarse feel for what was the architecture there. So you might love this or you might hate this. I'm open to other suggestions for coarse ways to look at this, but we just sort of looked at triads of neurons and how they were connected, these motifs, and their prevalence in these different areas. So these are basically representing the 3 different pathways, although they're not every single neuropil passed through along a putative feed-forward path through them. And sort of what stands out immediately is that if you look at this these ventrolateral neuropil pathways that are kind of the most direct that I'm going to talk to you about, sure enough, motifs that you might categorize as more feed-forward, so having fewer loops, at least within these triads, are much more prevalent in that area of the brain than, say, compared to, right, in dark black here, the mushroom bodies. Right? And so as you go towards what may be considered more recurrent or feedback type pathways, those rise in these other areas. So there are these different kinds of architectures as you go to different parts of the brain. And to the extent that we want to cartoon it at this point, the pathways I'm going to tell you about now have more of a feed-forward feel in their architecture. Yes?
Were you able to look at cycles of different lengths?
Yes. We haven't done that in the brain. I will get to that at the very end in terms of the nerve cord. I can show you one plot. And there are some differences too as well. Yeah, so I'll show you that data at the end. Yeah.
Just to get a scale, this is just shown in fraction of motifs, but this depends, of course, on the absolute number of these things. So what scale are we on that this will determine the differences here?
Yeah, that's a fair question. So I'm trying to remember, like the ventrolateral protocerebrum, I can't remember exactly how many neurons it has, but these are all probably roughly comparable in terms of like, to an order of magnitude in terms of how many neurons they have. But that's a good question. I should look that up exactly. This is good. Other questions? Okay. So let me dive into this first story of trying to understand something about the processing through this ventrolateral protocerebrum part. And fortunately, there's the pie charts. Fortunately, Srini also did a great job of introducing the fly visual system. Right, so you've seen this image before. Just to remind you, flies have compound eyes, which means they have, Drosophila have on the order of 700 different lenses, or we call these ommatidia. They come through the columns. And this columnar structure is preserved throughout these 4 different neuropil in the optic lobe, which means that you have retinotopic information as you go across any layer, right? So photoreceptors will send information down a single column. And we're going to look at neurons that have dendrites mostly in the lobula back here, some in the lobula plate, that have narrow dendrites that span a few columns but don't go across the whole neuropil, so they're getting sort of localized information. And these are the LC neurons, which you also just heard about in the last talk. And they have this sort of this kind of morphology. Basically, here is a particular LC cell type, and each of these individual lines here is not a dendrite, but actually representing an individual neuron. So a given LC type might have anywhere from 50 to 100 members in its population, and these individual neurons are distributed. They tile this retinotopic space across the lobula in the optic lobe. But then a given cell type, a given LC cell type, will send its axons into the central brain to a particular location. We call an optic glomeruli because they bundle. And so that's another way that you can distinguish these cell types. These colors here are showing you the actual axon bundle or optic glomeruli from the other sort of 20-odd of these LC cell types, right? So you have this sort of map of different optic glomeruli right here in this ventrolateral neuropil area. Here are some images of what these actually look like. So here's a population, for example, of LC18 neurons. And then we should be able to find here's an individual LC18 neuron. And there are about 20 of these LC types. In this particular experiment I'm going to talk to you about, we surveyed 10. The question being, what do these LC neurons respond to? What are they representing? They're visual projection neurons are coming out of the optic lobe. They're already 3 synapses deep in processing, so probably they've done a little bit of higher order processing. What do they represent? And so Nathan Klopotki, Alyosha Nern, who's in Jerry Rubin's lab, generated these beautiful GAL4 lines, split GAL4 lines. These target these individual cell types, meaning we can genetically express some kind of marker. In this case, it's a green fluorescent protein, or what I'm going to show you next is a calcium indicator. So we can go in and cell type by cell type, and in fact, neuron by neuron, interrogate their responses to visual stimuli. Nathan Klopotki was the postdoc who did this jointly between my lab and Michael Reiser's lab. And so what did Nathan do? He took the cell types for which we had nice clean driver lines, and he isolated regions of interest that were individual axons, and then he's going to center his visual stimulus on, for a given ROI, on the receptive field of an individual neuron. And then he showed a battery of motion stimuli derived from the literature, derived from what we already know about sort of earlier processing of the fly visual system that kind of cover the basis for all the basic kinds of motion that the fly might see. He showed about 100 different stimuli. I'm showing you kind of a subset here. We did looming, both dark and bright looming, small objects moving of different sizes, and of course moving in different directions and different velocities, small bars, thick bars, and then we had one wide field stimulus that was a grating. And so here's what he saw. He did see, in fact, that different LC cell types were tuned to different types of motion stimuli. You can see these do not have all the same response. And then if you look across a row, so for a given cell type, there were many that actually were quite specifically tuned, right? So one I'm going to talk to you about a little bit more is LPLC2, which is very tightly tuned for these dark expansions, so dark looming. But there were other ones like LC18, which actually were incredibly responsive to these small moving squares, which were actually sub-pixel resolution. So the Drosophila eye has about a 5-degree spacing between its ommatidia, and you can see these were 4.5-degree objects moving. So really interesting kind of array of responses here. The first thing we noted was that there was actually kind of a general categorical division. So there were a set that seemed to respond very strongly to small objects, be they dots or bars. And then there were this set down here that all had very robust responses to looming. So I had already kind of, given away this part of the punchline earlier when I was comparing to the Lucas work, but this blew our minds a little bit because, again, from the paradigm of, you have a looming detector, and then you hook it up to some kind of motor pathway you want, the idea that half of the feature detecting neurons in one part of the brain would be sensitive to looming was confusion and a surprise. What's more, if you look at the glomerular arrangement, so the actual spatial arrangement in the fly's brain, and how these different cell types were distributed, we find that the ones that respond to small objects are kind of all located spatially together, and the ones that are responsive to looming are all spatially located together. So we get kind of more of a conceptual feature map here in the central part of the brain compared to the dendrites of these neurons, which are in the optic lobe, which are retinotopically arrayed. Okay, and you can see that here too. So if you look at the similarity of their responses, the neural responses, compared to the distance between sort of the centroid of the glomeruli, you can see there's an inverse correlation. The closer they are, the more similar their responses were. And we presumed, or one hypothesis, I should say, for that, the reason that you would have this arrangement is, of course, to minimize downstream wiring. So you can imagine if you wanted to combine signals from a couple looming detectors, it would be helpful to have those nearby. And one of the beautiful things about the connectome, to bring things back to that and how we used it, is that I think previous types of studies have been able to survey cell types in a given region of the brain, but it's rare unless you have a connectome that you can then just arbitrarily ask, okay, now that I know the tuning of all these neurons, how are they connected downstream? So we're able to go one more step downstream and just look at postsynaptically who are the neurons getting input from these LC visual feature detectors. And one of the interesting things we noticed, if we just looked at, for a given downstream neuron, how many different LC types did it integrate information from? So the most prevalent was a single one, so single channels. And if we look at the morphology of these single ones, they tended to be local neurons. So there's some kind of local processing that's going on. But then by far, the next most common motif was to have a downstream neuron integrating from two features. Right? You can see that it actually drops off a lot by the time you get to 3 or 4 or like even more. It was very rare. So there's something that's very common about downstream neurons, especially downstream neurons that are kind of project somewhere else in the brain, specifically integrating from 2 of these types of features. And so here's some kinds of examples of particular LCs and their combinations onto downstream partners. Maybe a downside of the connectome is, right, of course, there's so many neurons that they get these arbitrary names that are very hard to remember and mean nothing, but that's why we draw cartoons. And then Nathan made a very nice matrix of all of these combinations to kind of look at which LC neurons tended to feed into the same downstream partner. And what you can take from these hotspots are that there's hotspots like these big ones where these are all looming sensitive neurons, right? So these are pairs of looming sensitive neurons converging onto downstream neurons. And then you had the converse, which are the pairs of small field or small object neurons, rather, and you get a very few of neurons downstream that are taking information from both a looming detector and a small object. Okay, how can we then use this information to actually understand something about how behavior is generated? I'm going to take you through the example of one particular pair we've studied a lot, which is this picture, LPLC2 and LC4. So here's LPLC2 and here's LC4. So we're now going to look into this box here and look at a particular downstream neuron. In this case, it's a neuron called the giant fiber. And so let me tell you something about the giant fiber, because the giant fiber, being the neuron in the fly's nervous system that has the largest axon, is actually was a pretty well-known neuron. There's two of them, a left one and a right one, a bilateral pair. This is what they look like. And it had been known, not through optogenetic means, but through means of just flashing lights on and off to white-eyed flies, that activation of the giant fiber causes the fly to do a takeoff. Okay, we can recapitulate that now using modern optogenetic tools. So we express CS crimson, red-shifted channel rhodopsin in this giant fiber. And sure enough, when we turn on a red light, that is what we see. Play that one more time. So we see the fly take off. So we knew in this particular case that there was a link to behavior, there was a takeoff. But what we didn't know was how that was actually used in the life of the animal, right? So we know that if you arbitrarily activate this neuron, it makes the fly take off, but that doesn't necessarily mean that that's what's happening when it's being attacked by a predator. So we wanted to then step back and actually try to understand if this neuron had any role in this behavior. We had a very intrepid student who actually went out and caught damselflies from the Genelia pond, put them in a cage with these dark smudges, which are Drosophila, and then we filmed these in high speed to see what the Drosophila would do when they got attacked. And we're incredibly relieved to see that our inbred lab-reared Drosophila could escape natural caught predators. So giant fiber, potentially still doing something. Just to build some intuition for you, I think everyone is pretty familiar with what looming stimuli feel like, but it feels a lot, right, like a baseball getting thrown at your head very suddenly. So that's what the fly is experiencing, and we're wanting to know, how does the fly respond to that? So we took the measurements we made from those high-speed videos, and we translated them into a series of looming stimuli that had the same expansion kinetics. And then we built this contraption we called the fly pez. The outside of the fly pez is a domed screen onto which we can project videos of these looming stimuli. Inside, we have a system of gating an individual fly onto a platform. We can use a mirror to see it in 2 views. And then we can record the fly's response to that looming stimulus at 6,000 frames per second, so we get sort of every detail of what happens. And then the nice thing about this machine is it's semi-automated, so it will let the next fly out after it shows this trial. And we can go at the rate of about a fly a minute. So we can get thousands of these trials per day if we want, which is helpful because even though, again, going back to kind of the straw man here of thinking of these looming escape responses as very stereotyped, if you start to look at hundreds of these, you see that in fact they are different. This is just a set of responses in which the fly actually took off. We actually have many responses in which the fly does other things, which I'll show you in a second. But even from this, you can see, right, this fly put its wings up really early, those a little bit later. That one did it one at a time. So there's all these subtle differences going on. And that's even in the context of the fly having many different behavioral choices it can make, right? So the fly sometimes just turns away from the looming stimulus. It might raise its wings up, and it might take off. And I'm not going to talk to you today about sort of our current and future work, but just to say that the ratio of responses of any given kind, whether it turns away, raises wings, depends a lot on the context, right? So we're now doing a set of experiments where we can starve the fly and put it on a patch of food, show the looming stimulus, and sure enough, right, the takeoff rate goes way down, right? So you have this variability, and it's probably for the fly to be able to kind of best adjust its response to its given circumstances.
So have you looked at how was the response of the giant fiber based on these different tasks? Have you actually looked at like sequence you're firing?
Yes. Okay, perfect lead-in. Thank you for the introduction to my next slide. Oh, one more thing, and I'm going to get there exactly. Yeah. Let me know if I don't answer your question, though. So before we get there, let me just point out one important detail, which is that when we looked at the takeoffs at 6,000 frames per second, so I will just emphasize that electrical transmission across neurons, across networks can happen at the millisecond time scale, so you kind of need to look at behavior at the millisecond time scale. What we saw was that, in fact, there were two different styles of takeoff, two kind of different motor programs. One that looked a lot like what I showed you before when we activated the giant fiber, which is that this fly is going to jump up and it's not going to have its wings up and prepared. And then it tumbles in the air, so it's sort of less steady. And then this one where the fly does a kind of very nice, elegant, balletic takeoff in which it has a very nice controlled flight afterwards because it prepared its wings. And you can see, if we just look at the duration, the time between when it started to raise its wings and when its tarsi leave the ground, right, we get this bimodal distribution that corresponds very directly with whether or not it put up its wings. Okay, and so then to get to this question, what role is the giant fiber actually playing in any of this at all? What is the giant fiber doing in response to a looming stimulus? And so in order to answer that question, we are going to do some electrophysiology. So we can head fix the flies. This is a fly that's looking at you. It's fixed to this plate. And we can insert a glass electrode on the opposite side of this plate through a hole. We can express green fluorescent protein in the giant fiber using genetic tools. And so we know precisely what neuron we're targeting. And then we can show a looming stimulus that's shown here in the red trace, which is just showing you the size of the circle on the fly's eye over time. And then here is an example trace from the giant fiber. And what you can see is that in response to the looming stimulus on this particular trial, the giant fiber fired a single spike. And so in general, I'll say that those are the types of responses we get. We either get just this subthreshold depolarization, or we get a single spike. And I'm going to play this as a video now. There's the giant fiber. And you'll see what happens when we get to that single spike. It corresponds exactly with the takeoff. So this makes sense with our activation experiment, right? When you activate the giant fiber, and in fact, all you need is a single spike, then it drives this takeoff. And I'm not showing you the behavioral data, but we could actually measure the timing of the wings going up in this head-fixed platform. And we saw something interesting. We saw that we always got a spike when the fly took off, but sometimes we would get a spike when the fly raised its wings and then took off, right? So it did this longer mode thing as opposed to just the short mode. When we activate the giant fiber, remember, we only ever see the shorter version. So that led us to try to understand a little bit more about what was going on here. And we went back to, right, this observation we had made from the connectome that all of this looming information, the only direct paths it has into the giant fiber we know from the connectome are through LPLC2 and LC4, these two visual projection neuron types. So that let us do this kind of experiment where we could record from the giant fiber, but then we could use our genetic tools to silence either LPLC2 or LC4. And then the way we're going to interpret this, again, leaning heavily on the connectome information, is that in the cases where we've silenced LPLC2 and we're recording from the giant fiber, we are going to interpret its response as coming purely through this LC4 channel and vice versa. Okay, so now this is a single example. I'm going to show you an average responses. So in black here is the average giant fiber response to looming in an intact fly, wild-type fly. And then here are the cases where we've silenced either LPLC2 or LC4. I've labeled them the converse, right? So for LC4 input, we silenced LPLC2. And the first observation is that they're different from the wild type. The second observation is that they're different from each other. So remember, coming into this, I kind of coarse-grained it for you, and all we really thought about LPLC2 and LC4 is that they were both some kind of looming detector. And this is already giving you a hint that, in fact, they're different kinds of looming detector. They seem to be providing different information to the giant fiber. So I'll try to make a long story short, which is that thinking about this, we were interested in what different optic variables might be corresponding with these inputs to these different channels. And we made the observation that if you take the size of the stimulus on the fly's eye, right? Remember, this is how we're showing the looming response for fast looming or slow looming, and you simply take the derivative of that size information, right? So now this is just how rapidly that size is growing on the fly's eye. And then you overlay that with this LC4 channel input to the giant fiber, the response to the giant fiber when it only has LC4. You can see it's a pretty close match. We can do that a little bit more quantitatively. We can now overlay the responses of the giant fiber with only LC4 input for different speeds. So these different colors of lines are now the fly responding, or the giant fiber responding to different speeds of loom. And you can see it doesn't matter whether it was a slow loom or a fast loom. The response through this LC4 channel pretty directly matches the angular velocity. This is on a log scale, so this is actually just a linear correlation here. And we can try the same analysis with the other channel with the LP-LC2 input. You can see we do not get as nice an overlay for angular velocity. We have to pick a different optic variable. And in this case, we just went back to directly the size of the stimulus on the fly's eye. And now you can see, again, different colors represent different speeds of looming, so different sort of speeds of predator attack. And you can see that these overlay very nicely. So it doesn't matter what speed the animal is seeing the looming. This LP-LC2 channel is pretty faithfully reporting the size of the loom. Okay, we can then put those into a model. We can basically, so here's like basically a closed form equation we can write, right? Representing the transformation between some kind of looming optic variable onto the membrane potential of the giant fiber through these different channels. It turns out, and I will show you some evidence for this in the next slide, that in fact, these two computations, these two looming computations, are ones that are found in many other brains where they're called either row cells or eta cells. So these are actually kind of common looming computations. And we can now build a model where we just add a row computation and an eta computation together for a given looming profile. And you can see when we add those up, we very closely match the giant fiber membrane potential. I will say there are two very small inhibitory components that I'm not showing you here, one of which is sort of feed-forward inhibition through LC4, but they're pretty small. So largely, we can understand how these optical variables are exciting the giant fiber. Okay, and then this is just to follow up what I said a second ago, right? These row and these eta cells have been seen in many different organisms, including mouse, cat, pigeon, and in several invertebrates. So to put it together in a kind of cartoon model, this is how we think the system works. And we simulated this in the computer, but sometimes it's just easier to show the cartoon. In the case of a slow predator attack, we think only the size channel through LPLC2 really comes to threshold. You don't get a spike in the giant fiber, and that there are other descending neurons coordinating that longer takeoff with the wings that then get activated and drive this other motor program. Whereas in the case of a really fast predator attack, then these two looming channels are co-activated. That's enough to drive that single spike in the giant fiber, and it's fast enough that it sort of beats down the pipe, this other information, and you get this short mode takeoff. So that's to say that in this system, what's really critical is the relative spike timing in these different descending neurons that we think are involved in coordinating these behaviors. And that spike timing is set by the particular input functions, visual input functions to these descending neurons, right? And then once we know that, it's kind of not a mystery to predict for any given kind of visual looming stimulus which kind of takeoff you're more likely to get. Okay, so we can cartoon this like this. So I just told you a story about now going all the way through the visual system from photoreceptors through these visual projection neurons onto the giant fiber and down into the motor side to get all the way to the behavior, and how how these two particular visual projection neurons might have particular looming functions that we can derive. Any questions there before I go on to second example? Yeah.
Yeah, I was wondering about the possible differences between individual flies. I mean, there's one that tend to jump and fly, and ones that tend to just spring their wings and fly. I mean, is that something that, or is this very stereotyped?
That's a great question. So we are now doing a series of experiments, which I won't talk about today, but we have some data where we recapture the flies. So in this set of experiments, it was one loom per fly, and then they flew away. But now we have a way of recapturing the flies so we can measure them repeatedly, responses to multiple looms. And what we see is that for the hour or two of that experiment, in fact, you do see some biases. So a fly that did like a short mode takeoff to loom 1 is more likely to do a short mode takeoff to loom 2. So they actually do seem to have some individual preferences in that way. But then if you take those flies, you put them in the vial for a night, and you put that same fly back in the apparatus the next day, there's no correlation with their responses to day one, right? So we think that it's not so much that there's personalities to flies or sort of any biases, but it's right, exactly. Right, no, it's state, right? There's these states that kind of exist on the scale of an hour or two, and those heavily bias the probability of a given response so that flies are consistent within that time, but then they don't persist longer than that, yeah. Okay, so the second story I want to tell you is kind of about orthogonal information that the fly needs to adaptively perform a kind of escape from predators, which is the fly needs to figure out where the predator is coming from and plot an escape route. And I will just say that there is a lot of, not a lot, there is some amount of theory on escape routes and like what is optimal, and this idea that maybe you don't want to be predictable if your predator can catch you, and so you might want to go somewhere random. That does not seem to be what's happening in the case of the fly. I will show you in a second some data. It seems that because this is so rapid and fly predators are very ballistic, that they don't need to be unpredictable. They need to get away from the attack as much as possible. You can just imagine the frog tongue extending. The frog tongue's not going to bend mid-flight just because the fly flew away from it, right? So its best chance of escape is to get as far away from that projected place as possible. Okay, but this is actually a very general problem, this idea of how we detect something in space, especially visually for us, and then command our bodies to do things. This is a sensory motor transformation problem, and you have encountered this probably during this talk. If you got thirsty, you see the bottle on the desk, you really want to sip. What you have to do before you can grab it, though, is you have to interpret the bottle's location in terms of where it is on your retina, and you have to translate that into some kind of command in body-centric space to actually extend your limb in the right direction. And so how do we do this? How are flies doing this? Can the Connectome tell us some of the secrets? And this is work that we did in collaboration with Mark Dombrowski and Larry Zapersky, who are here. And Mark also can tell you even more about this and sort of extensions of this work on his poster. I highly recommend stopping by. Okay, but so just first to show you that flies, in fact, do escape in different directions. So here is a video of a fly on a platform. It's going to experience a looming stimuli from the left of the screen. And let's look in detail what the fly is going to do. It stops grooming. It moves those middle legs forward. It's then going to extend its wings in this case and jump off the ground. So there's a part of this I didn't tell you about before where the fly actually adjusts its posture prior to executing the takeoff. And it turns out that that postural adjustment depends on the orientation of the fly relative to the stimulus. So now here's stimulus is still coming from the left. Right now that's on the side of the fly. And this fly, instead of moving its middle legs, does this very nice lean over away from the direction of the stimulus and then flies off the platform. And so this adjustment happens, and we just look at the timing of when things happen relative to the takeoff, which is here on the right. These kinds of T1, T3, or T2 leg motions are happening sort of in the first few hundred milliseconds of seeing the stimulus, but easily tens of milliseconds before the takeoff happens. Okay, and then just to show you that actually this is relatively precise, we can look at the location of where the fly, these adjustments are going to land the fly's center of mass relative to its two middle jumping legs after those postures have been done, but right before the fly takes off. And this color code is just to show you that that center of mass location corresponds pretty directly with the direction the fly is going to go. So there seems to be some kind of targeting of the center of mass to a particular location away from the looming stimulus in order to go the correct direction. I have all sorts of cool movies to prove to you that animals do this too. I'm going to have to pick one. I will just show you the ninja rat. Right, so jumping away from the predator, especially a ballistic strike like a snake, is something that even mammalian systems do, including us. Okay, so where do we look for this? Well, luckily we had information from the Connectome. This is coming back to 4 of those LC, these visual projection neurons that I told you were responsive to looming. I told you the first story about this pair onto the giant fiber, but in fact this is a whole network, as I alluded to very early in the talk, and there's a whole handful of other descending neurons that get this looming information very directly, right? So these are direct synaptic connections, including ones like DNPO4, where it's getting a large amount of information from LC4, which, if you remember, was the looming speed channel. Okay, so we decided to focus in on these descending neurons to understand if they had any role in any of these escape behaviors. And of course, I wouldn't be telling you the story if they didn't. So here is us optogenetically activating DNP11. And what you see across this whole set of flies is that when you activate this neuron, in fact, the flies lean forward, and then many of them are going to take off forward as well. In the case of another one, DNPO2, if we activate this descending neuron, now the flies are going to all lean back, right? So now instead of seeing the ends of the abdomen, you're seeing in green, which is the first frame, their heads. They're all moving back. Interestingly, in the case of DNPO2, the flies never take off. We have to co-activate DNPO2 with another neuron, DNPO4, to actually get the takeoff. And then here's just the quantification of that. So here, DNP11 causing the flies to lean forward, DNPO2 and PO4 co-activated, causing the flies to lean back. And then here were our controls, where they didn't lean so much at all. Okay, I'm going to get in 5 minutes. Okay, perfect. I'm going to show you now how we think this information gets transmitted from these visual projection neurons, especially LC4, that provides input to DNPO2 and P11. And one thing that I didn't highlight before, but it's kind of useful to understand, we can probably see it before, is that, remember we talked about how these dendrites of the LCs are retinotopically distributed, but their axons bundle. So in the field, the idea was that, well, retinotopic information is probably being thrown away because you don't have a really nice large space for a spatial map here. And in fact, if you look kind of at a coarse level, it looks like these dendrites for these descending neurons invade most of the glomeruli. So there's not a too strong and obvious bias for most of them, although I will say asterisk, C mark for all the details. But it turns out that if you look not at the spatial location of dendrites or axons, but actually now going back to the connectome at their very specific connectivity, we find that even though LC4 neurons we think of as a single cell type, individual LC4 neurons here on the x-axis are not making the same number of synapses with this downstream descending neuron, right? So here's our 55 LC4 neurons. You can see some make like 2 synapses and some are making nearly 60. If we look at the same kind of connectivity for LC4 neurons, so these same LC4 neurons, but now onto this other descending neuron, DNP11, we see the opposite gradient. And so now I'm going to draw a picture for you of what these gradients look like in terms of where the dendrites are. So I'm going to color each dendrite by the number of synapses made by that LC4 neuron onto either of these descending neurons. And we'll look in this orientation of the fly. And what you can see is that for DNPO2, which makes the fly lean backwards, you get a hot spot of connectivity or a gradient towards a hot spot at the front of the fly and the converse for the back of the fly. And so, okay, let me make sure I squeeze in the next part before I wax too poetic about this. This is, of course, just from the connectome, and I know we've been talking a lot about, well, are these synapse numbers really functional, right? This is a number taken from a dead fly. What could it really mean? So we went to the trouble of actually doing the electrophysiology to try to measure that. And so here's what we did. We had looming stimuli. In this case, we're trying to understand whether the downstream neuron here, right, it responds more strongly to loom in the frontal field than the hind field. So what we did is we arranged a column of looming stimuli to kind of try to create a really strong looming stimulus that looked like this in different azimuthal positions. We did that for both DNPO2 and DNP11. And long story short, here's average traces where you can already kind of see the answer. These synaptic gradients in the connectome are borne out functionally in terms of how much response you get in the downstream neuron, right? So for DNPO2, it does in fact respond with more spikes to frontal stimuli, and the reverse for DNP11, and that's true whether you look at spikes or whether you just sort of look at the area under the curve, because we're recording intracellularly. Okay, so just to put back up this sensory motor transform, and to say that, right, using the connectome, we can now just look at it and see something that was a bit mysterious before, these kinds of transformations between coordinate systems. It looks like this gradient, but it also is important, the architecture of these specific visual feature detectors being hooked up directly onto these descending neurons, which then drive particular actions. So that combined, that is the architecture that creates this particular sensory motor transform. And we think that this is how you control the fly going forward and backward. There are two DMP11s, one on the left, one on the right, two DMP02s, one on the left, one on the right. And so between the combination of those 4 cells, we think that that's how you could potentially have the fly control all 4 directions. And then just to highlight, because I know we've had some discussion about how many synapses and an important connection, right? These gradients go all the way down to single synapses. So on some level, individual synapses can have meaning. That's not to say that if we had done a coarse-grained look and a threshold of those out, we wouldn't have also still seen these patterns. So, I think it's just useful to keep in mind that they're important, but we might be able to find them other ways, and we should follow up our initial analyses. Okay, I'm going to stop there because I'm seeing the 0 number, but I'm happy to take questions. And while we take questions, I will just put up this slide, because there was also some questions about how accurate the neurotransmitter prediction was, and we tested that as well in the ventral nerve cord. And so what you're seeing here is from fish, basically like in situ hybridization of fluorescent probes with RNA for different neurotransmitters. And so this is the predicted neurotransmitter, and then this is what we actually found. So evaluate that as you will. I'm happy to take questions.